Alright, today we will be starting with the first chapter of the syllabus, which is seven. Whoa. Okay, so right, we or organisms all around us are generally the ones are very complex. But right, the question is, how do we manage to, for example, this particular device is a bit annoying. How do we manage to, for example, absorb nutrients or carry out gas exchange? Right? And the reason why these questions come up is because, right, gases, right, are either atoms or molecules, and right, the way these guys move is generally, right, for example, via diffusion. That is something we will cover, right? But if you have any idea of what diffusion is, hmm, hmm, that's not frequent. Actually, I'm on a different device at the moment, and okay. So they move through diffusion. And one of the things about diffusion is that it is effective over short distances, very short. Right? And so we as a complete organism, it's not like we are absorbing things through our hands. No, we breathe in, right? Air goes into our lungs, and right from there, oxygen into our body specifically, not everything else. And we eat food, and that is broken down into its component molecules, smaller molecules, and then it is taken to the body for nutrition. And that's not happening directly through the stomach, or food is being absorbed through our hands or our mouth. So there's something going on a very smaller level, on atomic level perhaps. And so when, right, with that, the first study and realization that we are made up of, a smaller fundamental building block was when a scientist named Robert Hooke, right, he studied cork cells. And he saw that the cork, right, so it's a plant material, and that cork was made up of tiny compartments, right, something like that. And because the plant cell, you can see the regular shape, but at this time, this was the first time they were seeing, oh well, you know, this is new. And he named them cells for a very different reason, but that's how they, he's the one who named them. Later studies, right, with microscopes, uh, gave us the, understanding that all living material. So he did this with lenses. So a proper microscope wasn't, hadn't been probably invented at that time. And so he was working with lenses and he studied this material cork. I can write it better on top. Yeah, so cork, C-O-R-K. And when he studied it under, high-powered magnification lenses, then he saw this structure. Later on, when other living material was studied, we, came to know that all living material is actually, made up of cells. And from there, the realization that, right, cells are the basic building block or basic unit of life. Why is this happening? Okay, I have no idea why that's happening. I have to ask you to excuse these glitches because, as I said, I'm on a different device at the moment. I'm not used to it, and it is apparently not used to me either. Okay. So cells are the basic unit of life. That's what this odd scribbling is. And cells contain cells don't contain atoms in the sense that they put free atoms of different elements or stuff like that, right? So cells are like an industrial complex, let's say. So, for example, in certain cities or certain regions, you have this area, right, where you've got a lot of different factories manufacturing different goods, right? So a cell is sort of like an industrial complex with many smaller factories inside of it carrying out different tasks. And each cell, we'll talk about specialization, and so, right, cells work together to form larger areas, right, of specialized function, and then even more complexity and so on. And that is how we are made, right? So we are just basically cells. We are trillions of cells that are working together. Now, before I talk about what cells are made up of, right, I want to go to magnification and microscopy first. So microscopy and magnification, right? So the light microscope was the first that was, invented, and pretty simple thing, right? It's got a place to keep a specimen. There's a light source below the specimen that shines light, right? And then there is a lens, right? So that light passes through the specimen, goes through a lens, is magnified, and then there's another lens, right? So there are two lenses, and then finally, you view it. So that's an eye, I guess, right? And so light microscopes can achieve a decent magnification, right? If you can magnify something up to, just a minute, yeah, you can magnify something up to 400 times, right? So you've got, there's a picture of the microscope in your textbook later on in the chapter, right? And there is the first lens that you have is an objective lens, right? And then there's an eyepiece lens. And so your total magnification of a specimen depends on the individual magnification, right, of each of these. So if, for example, the objective lens has a magnification of 40x, means 40 times, and the eyepiece lens has a magnification of 10 times, right? So the total magnification you're getting is 400x. And so that magnification is sufficient to be able to look at plant and animal cells, to, to be able to view them, not to be able to view all their details, just to be able to see the cells and, some of their uh, larger structures. And when you are using a microscope to, magnify or enlarge something and view it, then, right, there's a formula for that. So magnification equals to what should I call it? Size over actual size, right? And so if the image size, for example, is right, 2 millimeter, and normally what happens is that you'll be asked to find the actual size, right? And so you don't know the actual size, and that's x, and the magnification is, for example, times 100. And so you just move those things around, and so you can get the actual size, which is x, 2 divided by 100, which will give you 0.02 millimeters, which is pretty small. So the actual size of, a particular cell or whatever it is that you are seeing. Right, so this is one formula that you need to know. Perhaps as far as your O-level biology is concerned, the only proper formula that you need to know, perhaps. Any questions? All right, so this is a minor topic. This comes in later on in the chapter, but I thought about covering it first. And right, so the light microscope is what was used initially. Later on, the electron microscope was discovered. Details are absolutely not in your syllabus, but right, it allows magnifications up to, multiple thousand times. and with that, much finer structures that are not visible from the light microscope can be seen. Achha. Now let's talk about cells. Right? So generally, when talking about cells, there are three types. So you've got animal cells, plant cells, and bacterial cells. We'll talk about all three of them, bacterial cells briefly, plant and animal cells, right? And what we'll also talk about are certain features that we find in the cell, what the cell looks like, right? What does the cell have as its, what makes up a cell? So we'll look at uh, similar, or in fact, I think it's better to use common. So common features that are found in both animal and plant cells, and then certain unique, so to speak, features. Right, so let's talk about animal cells first. Animal cells, right, when they're seen under a light microscope, they appear as irregular shaped compartments, right? And what you can see is an outline, right? You can see a large dark colored blob in that compartment, and you can make out that, this compartment is filled with something, right? So that's all you can make out. And these are some of the, the larger structures that again are visible via the light microscope. And this outline or border is called the cell membrane. This stuff inside that is seen to be filling the cell is the cytoplasm, and this large dark colored blob is the nucleus. Right? And so let's talk about cell membrane. Right, so this is a thin layer surrounding the cell, right? It is partially, which is annoying, it is partially permeable, which means it is selective in the materials it allows to pass through, and also helps to separate one cell from another and and its surroundings. Right, now, why do you want it to be selective in the materials it allows? Because not all materials are wanted, not all materials are helpful. The things like nutrients that the cell needs to survive. Again, right, as we progress in the syllabus, we'll talk about that. But this is a very, very basic introductory chapter, so there might be questions. Oh, like, what do you mean by nutrients and, other stuff? Well, we get to that, but just as in an introductory lecture. So things that the cells require are allowed to come in. things that the cell does not need and they're found inside the cell, right? They are removed, so they are allowed to pass through. So useful things are allowed to enter, right? Waste products are removed, and the one cell is separated from, the other, and it's also separated from its environment, right? So there might be other things around the cell that don't belong inside, perhaps, right? Or the conditions outside the cell might be different from the conditions inside the cell. And so separating those two are important, right? Just like, for example, you have a net on your window, right? So you open the window, but you have a net on it. So you're allowing the light and the air to pass through, but you don't want the insects coming in because they're going to disturb, right? Or a lizard coming in, perhaps, right? Because if you see a lizard is going to be jumping around all over the house and it's going to disturb the working of your house, even temporarily. So you're going to put a net over this. So that net could be taken as an example for a cell membrane. Just an example to understand. Okay. Next up, let's talk about the nucleus. Uh, you can make, well, in that case, you will not perhaps see two different cells. What you see is, right, so this part might appear as one line, the part where two cells are joining together. But you will see an outline. So you perhaps, at least under a light microscope, you will not be able to tell two cells apart. You will be able to tell two cells apart, but you won't be able to tell the cell membrane in this part apart. You will see a membrane. You will be able to identify the membrane, but you can't say, oh, this membrane belongs to this cell, this part of the membrane belongs to that. It will appear as one. Okay? Okay. The nucleus. Now, right, the nucleus is sort of the control center of the cell. That's not a term that's given in the book. And right, it is responsible for, you know, again, controlling or coordinating cell function. So what's inside the nucleus is genetic material. Right, that genetic material is called chromosome. Right, chromosomes are DNA. Now we'll get to this all in detail later. But basically, what that does is, right, it uh, codes or regulates or again controls production of proteins, right? And these proteins carry out various tasks. So they're carrying out various tasks, and then depending on the type of protein, the type of task that is being carried out in the cell becomes has a specific function, right? So it is the genetic material in the nucleus. So it is the nucleus that gives the cell you know its job, let's say, right? What is it that the cell has to do? And then you've got the cytoplasm. right? And the cytoplasm appears like watery, jelly-like, and it is inside the cytoplasm where the nucleus is found, and as you'll see just a bit later, other structures of the cell are found. Okay, so these are the things about that can be viewed as far as your syllabus, the O level syllabus is concerned, that can be viewed under a light microscope for an animal cell. Any questions? If there's a problem with, well, it depends on what kind of problem it is. Uh, I, to answer your question very briefly, a problem with the nucleus probably means a problem with the DNA, the genetic material, right? So if it's the kind that can be repaired, it is. If it is the kind that cannot be repaired, then there is a check and balance, right? In the body, it detects that something's gone wrong with some cell in the body. Either the cell will trigger cell's death, so the cell will die on its own, or the immune system will find out, okay, there's a problem with some cells in a region, and the immune cells will come in and kill that, you know, malfunctioning cell, because the nucleus is, there's a problem with the nucleus, meaning there's a problem with the cell. In some cases, that malfunctioning nucleus, that malfunctioning cells survive, and that can give rise to cancer, right? But in most cases, it is taken care of. Either the cell dies on its own because of the whatever's gone wrong, or the immune system takes care of it. All right. Hey, what's your question? Well, the entire tissue is not affected, right? It's just one particular cell, so that's probably it probably dies and it's replaced by a new cell. You get, how it's replaced. We'll get to that later. So damage and repair is something that goes on. Again, that damage, the repair to that damage is also coordinated by the nucleus. Okay. Like for example, if you get a cut, right? So cells are damaged, but you have real life experience that the cut, the wound heals. How is it healing? Right? So it's not that the cut remains there for all your life, right? So yeah, and that is like physical damage, right? We're not talking about damage to the nucleus. That is physical damage. Okay. Now, right, talking about a plant cell again under a light microscope, when plant cells were observed, right, they were seen to have a very uniform structure, not as rectangular, maybe a bit more rounded at the edges, right? But they were seen to have a very uh, uniform shape. There was a large empty space inside, and there was a thick, or let's do that again. There was a sort of wall surrounding the cell, and then you got other things that you could see. So you have the cell membrane, which we just talked about. You've got the nucleus, and so you can see that these are common features. You've got the cytoplasm, right? And then outside the cell membrane, you have a cell wall. And then you've got this large sort of empty space, so to speak, a vacuum, right? So the cell wall and the vacuole are unique to the plant cell compared to the animal cell, right? So what do these two structures do? Mhm. So the cell wall, right, it is a rigid structure, right? It is made up of a certain compound called cellulose. So it is a rigid, rigid meaning it's not flexible. It is a rigid, structure. It is present around the cell wall, right? It is made of cellulose, right? Which is, we'll talk about later what that is. It's obviously a molecule, a very large molecule, right? It is not selective, right? So its function is not to check what is entering inside the cell or what is moving outside. In fact, its function is mechanical support. So it provides physical mechanical support by giving, a definite shape to the cell, and as we learn later, uh, you know, preventing it from bursting. And again, right, these are things we'll come to as we move along the syllabus. The vacuole is sort of, right, it's filled with, you know, watery substance called cell sap, right? It might also have ions and certain salts, right? Pigments are molecules that give color. Pigment, right? What else does it have? Yeah, and maybe, right, sugars as a store. And of course, the cell wall and the nucleus and cytoplasm we already talked about, so their functions are the same as in the animal cell. And when you see it under a light microscope, you would probably not be able to differentiate between a cell wall and a cell membrane because they're sort of, you know, pushed against each other, and they, right? So, right, they'll just appear as a thicker border around the cell, and you won't be able to tell them apart. Uh, yeah, and then yeah, one thing right, you will find smaller, right, structures which are called chloroplasts, and these guys contain the pigment chlorophyll. right? And this chlorophyll is involved in the process of photosynthesis, right? Which is basically how the plant makes its own food. Again, we'll talk about that in a later chapter, right? Using sunlight and carbon dioxide, right? And water. Any questions? Okay. So, right, if you then go into, a more detailed structure using an electron microscope, there are other structures that you would find in cells. So structures seen under an electron microscope. And we are given two of them, right? So one is the mitochondria, and it is said to be rod-shaped or sausage-shaped or slipper-shaped, or all these others. So basically an animal cell, right? You have the nucleus here, and you will see in animal cell and a plant cell as well, right? So in a plant cell, maybe they're made a bit more like the ones I'm drawing right now, right? And so if I'm able to yeah, so maybe they'll appear to be something like this. So this is a mitochondria, and another thing you would see, right, is a structure like this, and you will see these round objects attached to it, and maybe some of them just elsewhere in the cytoplasm, and these are called ribosomes. Right, now I've drawn an animal cell, but both of these are present in plant cells as well. So these are common features, common structures. And so the mitochondria is responsible for the process of cell respiration. And cell respiration uses sugars or glucose to provide the cell with energy. Again, we will cover this in more detail later. Right? And it is the process of using glucose, which is a sugar, to produce energy. Right? So the cell is working. It needs energy. Where is that energy going to come from? It's going to come from this process. Where does that process happen? In the mitochondria. The mitochondria is present in both animal and plant cells. Okay? And right, it has its own double membrane, right? Which means it has an outer membrane and an inner membrane, right? And the inner membrane has a lot of folds. So just something that for you to know at this point, like know this is, right? Without going into detail. And then you've got the ribosome, right? So the ribosomes can be either found attached to other membranes. Let's say to other structures in the cell, or right, found freely means not attached to anything, in the cytoplasm. And it is the ribosomes where protein synthesis occurs, right? So we talked about the nucleus controlling the production of proteins over here, right? So again, we will talk about how that works, not in a lot of detail, but we will talk about it. And so whatever happens in the nucleus then moves on to the ribosomes where these proteins are actually made. Synthesis means making, right? So these proteins are made at the ribosomes, and then they move on through the process and then go on to do their task. All right? Any question? Oh, yeah. In fact, let it be. So, all right, that's covered. There is a table in the book that you can refer to that brings all of this together. The the parts of the cell, structure of cells that are found both in animals and plants, their description, their function, and then the three structures as far as our syllabus is concerned that are only limited to or will only found in plant cells. After that, very briefly, right, we'll talk about bacteria, hmm? Organelle. Yeah, so it's a good thing you brought up that term, right? The mitochondria, the ribosome, right? And the chloroplast, what is that? This guy, right? And even the vacuole, the nucleus. So all of these, there's a term for that called organelle. And what organelles basically means is small organ, right? And so each organelle has a specific role. So remember the analogy that I gave of this industrial complex of the cell, right? So this might be the cell membrane, and this might be a mitochondria doing its job. This could be a ribosome, right? This could be the nucleus, right? And so you've got all these other compartments within the cell that have specific roles to play, and then all of them working together is what makes the cell do its job, right? But these cannot survive in, like, these are not taken as individual building blocks, right? So the cell as a whole, with all of its components, all of its structures, right, is taken as one, the smallest unit, living unit. All right. Okay, so it's a good thing you mentioned that. Now, right, it's a short topic, and with the time, what's that? No, go back to the board. Uh, Bacterial cell. So, not a lot to talk about these guys, right? Obviously, there are many different types, but we're going to talk about the general, right? So, what do they have, right? Bacteria, first of all, right, is plural, and the singular for that is bacterium. When you're talking about just one bacteria, right? And these are very small organisms. They're single celled. So unlike what we were talking about right now, animals and plants are like huge creatures, right? Bacteria are single celled. So all the features of the cell, well, I mean, not all, right? But they are like, right? They have a cell wall, sorry, cell membrane. They've got, in fact, they don't even have a proper nucleus, right? So this is genetic, I'm writing genetic material because they don't have a proper nucleus, right? And they've got in fact, they don't have mitochondria, right? They've got ribosomes, right? Some of them have certain other features, so let's just look at that. They have cell walls, which may be made up of, right, sugars, proteins, and lipids. So these are different biological molecules which we will talk about, right? And they have the cytoplasm. In the cytoplasm, they've got, granules like of food reserves. Granules means, small pieces, because, they can't go around hunting like us and, cooking food and having biryani and chow mein. So those food reserves might be glycogen, which is a sugar. It might be some form of lipid. Well, I shouldn't call glycogen a sugar, uh, right? Uh, then you've got ribosomes, and they're smaller than the ribosomes that are found in animal and plant cells, but the function is the same. Then they've got, so ribosomes are smaller but same function. And then, as I mentioned, they don't have a nucleus, so what they have is, right, circular DNA. So their genetic material is just present in the cytoplasm as circular DNA without a nucleus. All right. And then they've got other smaller pieces of DNA. Something wrong with the screen of this device. I have to obviously work with it at the moment. So these are called, I'll write it here, plasmid, right? So smaller circular pieces of DNA called plasmids. Nice. There is no nucleus in the bacterial cell. Uh, well, the main DNA in bacterial cells is the circular DNA that I mentioned, and it's just there in the cytoplasm, right, without a nucleus. and it does its job, right? So the functions controlling cell activities and controlling cell development and cell division. So when one cell becomes two, again, something we'll talk about later, right? So all of that is still controlled, and plasmids are, other smaller pieces of DNA also inside the cytoplasm. They do not have a nucleus, otherwise you would have more than one nucleus in the cell, and that's not the case. And the material that the plasmids have, help the bacteria to, survive and reproduce. And to reproduce, right? And the circular DNA, this guy is, I should say, regular cell functions, right? Development and division. Cell division means going from cell division biology means the cells are growing. They're growing in number, right? So in biology, division means multiplication. Okay. Mm, all right. So yeah, I think I'll stop here. And then from the next class, we'll talk about specialization of cells, and then, systems, so tissues and organs and all of that. And yeah, with that, we'll be done. And so, right, as I mentioned, we need to keep a certain pace because, right, there is material to cover. There might be some points that I missed mentioning, maybe, and so I do go over the text, and if there's any question later, right, just ask. So is there any question right now? All right, so I hope all of that makes sense. Apologies for the technical difficulties with writing, and I'll see you next time then, inshallah. Allah.